So now, on to the main event for tonight, to hear the Emeritus Professor George Yeoh talk. We're absolutely thrilled that it is George, his homegrown product. And I've known George for at least 20 years, George. More. We won't say how much more, but more. I've had the privilege of knowing George. I'm a lucky person. He's been absolutely wonderful at the Perkins. He's generous. He's humble. He's deeply committed. And of course, an extraordinary scientist. And we're going to hear about that tonight. George is the leader of the liver disease and carcinogenesis lab in the Perkins, and his pioneering work in liver stem cells is paving the way for new therapies to repair damaged tissue and to detect liver disease earlier, including the development of life-changing blood tests for children living with cystic fibrosis and liver disorders as a consequence. George is a twice National Health and Medical Research Council Fellow. He was the President of the Cancer Council of Western Australia. He's dedicated his career to advancing medical research that improves lives. His work reflects persistence, collaboration, and a deep belief in the power of science to serve the community. George was fundamentally involved with myself and Nina Turnitz-Parker on starting the biggest funding episode in liver disease in the nation, the Liver Cancer Collaborative, which is trying to beat liver cancer with as much power as we can. He's a generous mentor to young researchers. They flock to him. He's a wonderful teacher and wonderful mentor. And when he's not working hard, trying to find a cure for liver cancer, he is an avid, I mean really avid tennis player. And I think it's Raybould, isn't it? Is your club Raybould? Yeah. Corinthian Park, yeah. He's a mad dog tennis player, and he used to work on the Hopman Cup in his spare time. So, not much of that. So look, it's an absolute pleasure to welcome Professor, or Emeritus Professor George Yeoh to deliver the 2025 Harry Perkins Wesfarmers Oration, and the title is An Extraordinary Life and Adventures in Research. Please join with me welcoming Professor George Yeoh to the stage of the day.
Yep, you good? Okay. Okay. So there's been a lot spoken about me, but this evening's proceedings is not really about me. When I was asked to deliver this oration, I thought, great, I'll be able to talk about you, because what I have achieved is mainly the result of the generosity of the people of Western Australia. I'm so fortunate to meet good people, generous people, caring people who looked after me when I came to this state and this country. So I will tell you my story, but I want you to remember, bear in mind as I go through, that it's not about me, but the people that I will be mentioning who made it possible for me to succeed and to be standing here telling you about my research. So in my acknowledgements, I'm sure I'll miss some people, but if I say, thank you, all the people of Western Australia, thank you, all the people of this wonderful country, you have made it possible. So this really is an acknowledgement, a journey of acknowledgement. So I will tell you about my research, but along the way, I will tell you about all the help that I received. So I will start by my journey. And the illustrations are here, have been provided by my youngest granddaughter. And that's a picture of acknowledging her. I wrote this story, and Penny illustrated it because she asked me, Papa, where did you come from? How did you come to be in this place? So I told her, and I used that to her illustrations to tell you the story. So, in 1960, my mother decided to educate her 2 sons in Australia with the aim of them becoming doctors. She committed 1/3 of the entire family's wealth to this undertaking. The following year, we traveled to Singapore. We lived in Kuala Lumpur. We boarded the merchant vessel, the Karen. We sailed for 5 days. I got awfully seasick when we reached Sharks Bay. And then we arrived in Fremantle, and 2 Maris brothers picked us up and took us directly to New Norcia. I ended up with work being first term at St. Ildefonsus. We were enrolled at Guildford Grammar, but there was no space for us. But fortunately, after term 1, I then went on to Guildford Grammar School until I graduated in 1964. So at St. Ildefonsus, the Maris brothers put into me what it meant to work hard. They taught me Christian values, which I put into practice for the rest of my life. There were families who took upon them to be hosts. My Easter would have been very, very lonely if it hadn't been for the Clune family who said, look, you're going to be by yourself at Easter time. Why don't you come home with us? And this kind of generosity just really astounded me. People asking you, a foreigner, to come into their homes. Just think about that. So I went with the Clunes, had Easter with them, and Mr. Clune took me crabbing. And my English was very poor, but when a crab bit Mr. Clune, I heard a lot of English words that I'd never heard before. When I went back to St. Ildefonsus and I repeated those, I got the bat treatment. It's a cricket bat meeting your bottom for saying bad words. Well, I went on to Guildford Grammar School. And here I list the people who were instrumental in directing me. My housemaster, Mr. Macmillan, taught biology. He was my biology teacher, and he introduced me to microscopes, and I loved microscopes. So I went into looking at cells. Mr. Merriweather was my English teacher, who said, George, your English is atrocious. I'm going to send you to Mrs. Ward at Sawyers Valley, and you're going to go up there and learn to speak properly, learn to spell properly, and understand what English grammar is all about. And I spent most of my time arguing Mrs. Ward about English pronunciations. I thought, what is this forehead? It's forehead, not forehead. Anyway, I didn't get my way. My physics teacher, Dr. Greenhouse, taught me the value of science. Rigor, the scientific method. Students and families, and again, the generosity and the community spirit that was instilled into me held me well for the rest of my life. And I found that although I was pretty proficient with my academic ability, I wasn't that well accepted until I proved myself in the sporting arena, and what was absolutely necessary. So I shall digress a little bit and tell you about tennis. So I achieved full colors. I represented the school in tennis, and my tennis master actually appointed me as the tennis captain for the school in my final year. Through tennis, again, I received so much. Bruce Robertson was my tennis coach, and he told me he played at Wimbledon, and I was gaga when I heard about this. I became a member of the Corinthian Park Tennis Club and given life membership at the end of my tenure. I worked at the Hopman Cup from 1990 to 2011. Paul McNamee was my boss during the Christmas break. And you can see there in the tournament staff that I'm listed as in charge of match control. So we were in charge of providing food, making sure the nets were changed between the doubles and the singles, that the lines people's seats were in the right place, etc., etc. So I did this, and Paul's acknowledged this and thanked me for a job that I did for many, many years. And it was, again, a pleasurable break from the university when it came around. I'm very proud of this. I read a lot when I was at Guildford of tennis books, and this one, Aces and Places, was autographed by Lucy Hopman. Many of you will know she came to the Hopman Cup regularly. And she indicated there, God bless you, George, and Harry too, Lucy Hopman. And I had derived enormous pleasure from working at the Hopman Cup and contributing, putting back into the system, because tennis had given me so much. So that's a digression. Back to the work. My education and training in Australia. Well, the first person I came into contact with was an undergraduate collaborator, a graduate collaborator, a PhD collaborator, and a postdoctoral collaborator, and a friend. Max Cake, who's Professor Max Cake from the Murdoch University, we worked together. In fact, I have to confess, Max was my chemistry teacher. He tutored me in chemistry. That's the only way I got a distinction in chemistry. But here is the generosity. Not as somebody as a competitor, but as a friend. And he guided me. I then went and did a vacation scholarship with Will Simmons in the physiology department, who introduced me for the first introduction to research I had. And his technician was a Valerie Orton. Well, Valerie Orton became Valerie Yeoh. So once again, I derived enormous benefit from doing my research. My PhD supervisor, Professor Ivan Oliver, actually introduced me to the liver, and I was smitten. I was actually doing medicine. Then I switched to science. Well, you can imagine the shock my mother had when I told her, I'm going to leave medicine, I'm going to do science. But I would be a doctor, Mum. No way, I don't want that kind of doctor. I want a proper doctor. Too bad. So I switched. And then the head of physiology was my postdoctoral mentor and was very instrumental in research skilling. He taught me how to do research in a rigorous way, what were controls, how you design experiments, how you had to keep up with the literature. So my research interest was the liver. Now, the liver delivers. It's a vital organ, performs over 500 functions. I've listed some here: digestion, energy production, detoxification, blood clot, urea synthesis, and it's a travel axe of the body. It exchanges carbohydrates, proteins, fats, etc. So it is a very, very important. No liver, no life, okay? Many liver-specific genes have to be carefully regulated because we have a changing diet. When we are in a fetus, we're getting the blood from mum, postnatal, milk from mum, then we go to solid foods, and adults eat all sorts of things. And so it's very important to regulate these activities so that your enzymes can cope with the environment. At that time, I realized that because the liver was changing so much in that environment, one way to study it is to take the liver cells out and put them in a constant environment. Then, when the liver cells do things, you know if it's different from the fetus to the newborn and the adult, that it would be an intrinsic alteration in the cells, differences, not due to what the environment is telling it to do. So I needed to learn how to culture liver cells in a constant environment. And at that time, cell culture was in its infancy. I dragged Val off to the University of Pennsylvania in Philadelphia, and she said to me, "Why are you going to Philadelphia? I mean, can't we go to the University of Hawaii?" No. In Philadelphia, there was Howard Holzer, who was an expert in cell culture, in microscopy, and he studied how muscle cells were born in the body. And he studied how embryogenesis developed from a single egg, cell egg, to different cell types. So he coined the term quantal cell cycles. So he tells us that for the embryo to become an adult, cells have got to multiply, you've got to grow, but cells have to change so they make all the different cell types in the body. So that was important. That's why I went to the University of Pennsylvania. The Beatson Institute in Glasgow was a really rough town when we went there, and I still decided I had to go there because the cell culture specialist was John Paul. He was head of the Beatson Institute, which is a cancer research institute, and he had the best tools for cell culture. He wrote the book called Cell and Tissue Culture, and that was our Bible. So I went to John Paul and said, I wanted to work with you. So I got a C.J. Martin Fellowship from the NHMRC, and I went to Philadelphia and to Glasgow. In Glasgow, I learned again cell culture, instrumentation, because he was surrounded by people who were so smart, they made their own equipment, they made their own instruments. And there I studied how blood cells were born. Armed with all of this tissue culture expertise, I came back to Perth. I became an NHMRC fellow, and I then took liver cells from the embryo. I'll get the laser pointer, then I can show you. That's a little liver in the embryo, bigger liver in the newborn, and a large liver in the adult. So my idea was, now that I know how to culture these cells, take these different livers at different stages of develop, put them into a culture medium, and see what they do. If they do different things, it's because they are inherently different. And indeed, if you take fetal hepatocytes and put them into culture, they look like this. Take newborn hepatocytes, and they look like this, and adult hepatocytes, they're much larger, and they look like this in culture. So I had the tools now to study how the liver actually develops. What we showed was that the fetal hepatocytes, if we keep them in culture for long enough, they actually take on the characteristics of the newborn hepatocytes. So we have seen in this culture medium a conversion of fetal to newborn liver. So there is differentiation right before our eyes. But we also found out that it's not just a matter of time, the cells had to divide and grow. Because if we put in an agent that stops the cells dividing, the fetal hepatocytes do not become newborn hepatocytes. If we block DNA synthesis, we block cell division, that transition didn't happen. So here is the question, what is going on? What are the events that change a fetal liver cell into a newborn and a newborn into an adult? So these were the things that I was fascinated with. And for me to be able to address them, I had 3 different laboratories around the world who were generous enough to provide me with reagents. They had cloned these genes, they are a mouthful, tyrosine aminotransferase, phosphoenolpyruvate carboxykinase, and pyruvate kinase. One, the enzyme tyrosine aminotransferase was at the Deutsche Krebsforschungszentrum in Heidelberg. Beautiful place to work. And that's where I also ran into Ruth Gans, who's here in the audience. And I again acknowledge that wonderful group that I went to work with, generous with reagents, generous with passing on their expertise. I also went to Darrell Granner at the University of Iowa because he had cloned PepCK, short for phosphoenolpyruvate carboxykinase, and he provided me with antibodies, the DNA clone, and so I could measure the messenger RNA of this enzyme. And in Japan, Tamio Noguchi, who was at Osaka University, cloned this pyruvate kinase gene. So these were huge landmarks. They were the first time these genes were cloned, and these people had very valuable reagents. They could either keep it to themselves or be generous enough to provide it for me to take back to Perth so that I could do my experiments. So, to cut a long story short, we established that the liver repairs its damage by two methods. One of them is acute liver damage, so that if you get your liver poisoned by taking drugs like paracetamol or physical damage, then you use this, which I call proliferative cell cycles, which is what Howard Holt said. I went to Philadelphia to work with. So it's cell type H, which is a hepatocyte, divides and gives H and H. So your liver is a powerful regenerating organ. If you lose 1/3 of your liver, you'll grow it back. The amazing thing is it will grow back just what you lost. Not a little bit less or a little bit more, exactly what you've lost. If you're in a car accident, you've damaged 1/3 of your liver, 1/3 of your liver will grow back. So it's an incredible organ capable of extensive regeneration. However, the other side of the coin is that if you have chronic liver damage, so you've got to look after your liver will look after you. And we found that with people with alcoholic liver disease, iron overload from genetic hemochromatosis, or hepatitis virus infection, these type of divisions occurred. The liver stem cell becomes a cholangiocyte and a liver cell, or the stem cell becomes a stem cell and gives rise to a hepatocyte, or the stem cell gives rise to a hepatocyte and a cholangiocyte. And here, the stem cell gives rise to one of its own and another, which is the cholangiocyte. So these are very different kinds of cells that are growing, all in the culture dish. Okay, so it's not due to their environment. They are inherently different. We can conclude that. And another model that led to my interest in liver stem cells was, did the liver develop homogeneously? So all the cells at one stage in the fetus were all fetal hepatocytes. In the newborn, were they all newborn? In the adult, are they all adult hepatocytes? Or is the mechanism heterogeneous? So here you have a fetal hepatocyte giving rise to newborn hepatocytes, but it leaves behind a tiny amount of the fetal hepatocytes. And then when the newborn hepatocyte gives rise to adult hepatocytes, you still have a tiny fraction of the fetal hepatocyte. And this model is what I subscribe to by looking at these liver cells growing and differentiating. And this model predicts that even in the adult liver, in your liver, there are some liver stem cells, okay? So that's the concept of a liver stem cell. And then I received, again, more and more support while I did travel through my journey as a researcher. Head of departments, Evan Morgan, head of physiology, Norman Palmer, the head of biochemistry, welcomed me into their departments, provided the resources so that I could do my work. I also had a group of very, very supportive fellows, NHMRC fellows, Fiona Stanley, Roy Goldie, Lynn Beasley, and Jeff Shellam. I'll tell you a little story. We had to go to Melbourne to be interviewed for our extensions of our positions as NHMRC fellows. 3 years, we were told, yes, we'll reappoint you and extend your position, or we may not extend it, and then you find another job. And Fiona, at that point, was about to have a baby, and she looked at me and said, George, I don't think they're going to extend me because, you know, I'm not in a good place with, how can I do, they be convinced them that I'm going to be working effectively next year. I said, Fiona, with your reputation, don't worry, they'll extend you, right? And sure enough, Fiona was extended. But guess who wasn't extended? Moi. And I was a little bit puzzled, and I think it's worth telling you this story. Jeff Shellam took it upon himself to go back, because he was a wee high boy, so he was very much in contact with the people in Melbourne. He found out that I didn't pass the test for 2 reasons. One, my publications weren't in number sufficient, and in quality insufficient. But the more important thing was, When I went for my interview, I told them about we did this, we did that, and a lot of them scratched their heads. George talking about we. Is he talking about Evan Morgan and him? Is he talking about Ivan Oliver and him? So what is he doing as his own? I should have been saying I, right? So Jeff took upon himself to ask the panels to ring up Ivan Oliver and Evan Morgan and find out when George says we, is he actually referring to his own work? And luckily, both of them said yes. Right. So I wasn't extended that year, but when I went back the following year, I got the Guernsey. So once again, the comradeship of these people is amazing. You know, I mean, Roy Goldie and Lynn Beasy are firm friends of mine, and so is Fiona and Jeff Shellen. Faculty of Medicine Deans, Lou Landau and Ian Puddy, made me an Associate Dean of Research, a dropout from the medical school. Think about that. All right? They're prepared to give me the chance. And both of them gave me the opportunity to give back, to start new funding schemes for the faculty, fund new scholarships. And both of them said to me, George, go for it. If it fails, we'll tell you to stop it, right? But if it's successful, keep going. And so they gave me the wherewithal to do what I wanted to do to help the medical community. And UWA executives who really supported my research. Robin Owens was the Vice Chancellor of Research at that time, and Alan Robson. These two people were responsible for UWA getting into the top 100 of the universities of the world. Right? They really supported research. And Alan Robson in particular was instrumental. I wanted to run a meeting in Perth called Liver Down Under. And I did run it. He underwrote that meeting. And I would still claim that this is a meeting where we attracted 10 of the top liver researchers from America, the UK, and Europe. The top people came. We had Australians, the wise men from the East come over to the West to learn. Fantastic. So we ran this successful meeting, and it wouldn't have been possible if Alan Robson had not underwritten this. I've got a circle around a character here. His name is Marcus Grompy, and we'll come back to Marcus. So mutual support, institutions and executives. I've been involved with the Corinthian Park Tennis Club for a long, long time. I was their captain, president. I've been made a life member. And this is due to the president of Lawn Tennis WA, Alan Hicks, and his wife, Mary Hicks. Again, the opportunity to give back. The Cancer Council of Western Australia gave me my first microscope. And honestly, I was thinking of going over east or maybe going back to the US to do my work because it was tough going, getting grants. Clive Deverell was the first CEO of the Cancer Council when I got my first grant. The picture there is in black and white because they didn't take color pictures then. Hendy Cowan was the president, followed by Christabel Saunders, and now just past president is Ruth Sheehan. They all supported me. Suzanne Rooney, who's the CEO, and Ashley Reed, the current CEO, and Terry Slevin was the research supervisor for the Cancer Council. So I'm indebted to them. I got also involved with the Institute for Respiratory Health, and for a while I was the director of the Center for Cell Therapy and Regenerative Medicine. And this was very much due to the efforts of Jeff Laurent and Jeff Stewart, who invited me to join them. The Lions Eye Institute, I'm a researcher on the Research Advisory Committee, and two people, names you'll know, Ian Constable and Bill Morgan, who actually helped. And Bill is the son of Evan Morgan, who was my first mentor. St. Catherine's College, of which I'm a fellow, Fiona Crowe invited me to organize the fellowship there. And it's great because you meet the young kids who are in the college, talk to them, and they're enthusiastic about what they're doing. And of course, whenever I have an opportunity, I'll tell them how important the liver is. Royal Perth Hospital Research Foundation, and there are people here from that foundation. I'm their research strategy advisor. They listen to me. People involved, Lynn Beasley, John Palermo, who's the current chair of the board, and Kate Kasakis, who's the CEO. So once again, they've been instrumental in allowing me to give back, which is so important for me because I just keep feeling that I'm still on the wrong side of the ledger in trying to make up for what I've received from this wonderful place. The Lions Cancer Institute, I'm a joint patron, and the other patrons are also in the audience. His Excellency and Mrs. Dawson are patrons of this great foundation, and they are now supporting liver research, as well as melanoma research. Gary Irvine and Phil Chinnery were instrumental in bringing me into that. So a lot of giving, but a lot of receiving that I've had from all these people. I also got involved with societies. I was a local member for the Australian Biochemical Society. I ran meetings, the Australian Society for Medical Research, and I was a founding member of the Australian-New Zealand Society for Cell and Developmental Biology. This was started by Keith Dixon in South Australia, and I did the West Australian part. The Australian Society for Stem Cell Research, of which I'm a founding member, I am the first president of that, and my membership number is 0001. The Combined Biological Sciences meeting came about because Miranda Grounds and I were running meeting after meeting in Perth for the ABSM, the Australian Society for Medical Research, and the ANZ SCDPI. We thought, why run 3 meetings? Why don't we put it all together? So we founded the Combined Biological Sciences meeting, which still runs today. And they were good enough in 2015 to make myself and Miranda life members. So we can go to the meeting for free. Well, the 2 Peters, in 1980, Peter Liedman, that's when I was invited to join WAMR, the West Australian Institute for Medical Research. And my research efforts were significantly enhanced when I accepted this invitation from the 2 Peters, Clinton and Liedman, to join WAMR. And that's my badge from the Royal Perth. Look at the black hair there. So that was 1980 when I got that. I still use it when I go to the Royal Perth Hospital now. But they don't recognize me anymore. But it's an amazing research environment, WAMR and then the Harry Perkins. And once again, there are many of you here who are supporters of these institutions. You know who you are. Just think, what is the legacy that you guys are leaving behind in supporting and enabling the sort of research that's going on? Just amazing. And, as Peter told you, this will even be further strengthened when we have a comprehensive cancer center. We deserve it, and we're going to get it, all right? And that picture there is Peter Klinken, who I encouraged to go abroad and do research, and he was at the NIH. And I spent 6 months at the NIH, and he offered me accommodation when I was there, okay? We got into the semifinals of the NIH doubles tennis championship, and I had to go to a meeting, and Peter Klinken wasn't happy because we had to forfeit. But that little girl there is Elizabeth Klinken, and that red circle will come up later in my talk. Okay, so research at WAMR then, we focus on the liver stem cell. And when I treated rats with excess ethanol, they drank heavily. We gave them all the grog that they wanted. And interestingly, what happened was the ethics committee got to me and said, hey, George, we want to come and inspect these rats. We want to make sure that they're healthy, that you're not giving them too much alcohol, and they look like alcoholics that we see, derelicts on the street. So, well, come and have a look. And I tell you what, they came and had a look, and here are these fat, happy rats, drinking lots of alcohol, and they said, no worries, keep going. Anyway, those rats develop these, what we call oval cells, which are the rat liver stem cells. And when we chronically iron overloaded rats and gave them too much iron, these cells also appeared. okay? So there are two conditions, alcoholic liver disease in humans and genetic hemochromatosis, where they get overloaded with iron. And in both cases, I wanted to have a look at human liver, right? So we'll come to that. The most important thing that we developed here that we sent all over the world is that we developed a fatty liver mouse model. They had fatty liver disease, and then they produced these liver stem cells called oval cells. Here's the paper that we published. And what is interesting, if you give them this diet long enough and you insult the liver frequently enough and over extended period, the mice develop liver cancer. So here was the first link between these stem cells and liver cancer. And I'll return to that story. So I established these liver stem cells from these mice. And here's the paper that we published. I acquired this technique from Mary Weiss. I wrote to her and said, look, I'm trying to do this in Perth. I'm not succeeding. Now, I said, I'm skeptical whether you can do it as you say you can. Well, Mary wrote to me and said, George, you don't believe us? Come to Paris and work for 6 months. We'll teach you. And I went to Paris. I worked for 6 months, and I learned the technique. Here again is the generosity. So I came back, and we worked on these cells. But here is a very important thing. I wanted to grow lots and lots of these cells to use them to treat people with liver disease. We had to find out what makes them grow. And mice you can get that have different growth factors knocked out of them. And here I used these mice, and I showed with my diet that the fatty liver diet, it didn't allow these cells to grow. So I knew when the mice didn't have IL-6, if they didn't have TNF-alpha, lymphotoxin alpha, or this called tweak, which Janina Parker worked on, these were very, very important growth factors for these cells. So if you want to grow them, you add it to the medium, and they'll spring up like mushrooms. And we also had the link between these cells and the liver cancer. We found that if the mice couldn't make these cells, they was less likely to get liver cancer. So we proposed that the liver stem cells had a Jekyll and Hyde characteristic. They could be good and they could be bad. And so, to move into the clinical, the human setting, you have to find a clinician who's passionate about research like you are. And I found this person in John Olnick. John is dedicated, he's a passionate scientist, and he's a clinician scientist. He made it available, made liver samples from the Fremantle Hospital available to me, right? It's like gold. So we published this paper, and it's been cited 613 times. We've showed that oval cell numbers in human chronic liver diseases are directly related. They are there, and they are related to disease severity. Notice the mice that had iron overload, humans with genetic hemochromatosis with iron overload, humans who have alcoholic liver disease or chronic hepatitis C infection got a lot of these liver stem cells in their livers. And here's a picture. So I need to explain this. The blue dots are the cells. The black dots are the liver stem cells. And I was very fortunate again. Martin Perra at the Australian Stem Cell Centre made an antibody that marks these cells. We can mark them brown. You can see in alcoholic liver, they're scattered all over. In fatty liver disease, yes, they are. And in autoimmune hepatitis, indeed. So this was a very valuable reagent that we could use to stain liver samples to see how many of these cells and whether these cells were there. This is important because I'll come to what I'm doing today with the cystic fibrosis kids. So bear in mind, the blue is the cells, all the cells, the brown are the stem cells. Some of them good, some of them bad. So I merrily used the technique from the Pasteur Institute and made all sorts of liver stem cells. I made blue ones from a blue mouse, so blue liver stem cells, and I injected them into mice and formed blue bile ducts. I injected into mice and formed clusters of liver cells. So these cells can either make bile ducts or they can make hepatocyte liver cells, 2 types of liver cells. They can go either way. So we call them progenitor cells because they can at least have 2 options. I also made them for a mouse that had the jellyfish, the green fluorescent protein from jellyfish, inserted into the mouse. So the mouse now is like a jellyfish. It grows green, bright green. When I make liver stem cells from them, they glow bright green. That will allow me to transplant them and follow them. In this case, we actually transformed them into scaffolds, made them differentiate, and we can see them easily, as you can see here, that's in a scaffold. And the idea here is to put that scaffold back into somebody who's got a liver deficiency and see whether these cells can actually rescue the person. So now I was busy making all these cells, transplanting them, seeing what's going to happen. But I received 2 significant setbacks. Some of the liver stem cells that I sent to my collaborators called me up, sent me faxes and said, George, I've transplanted the cells, but my mice now have got tumors. They've got liver cancer. We've given them liver cancer. And then this guy, Grumpy, that was the guy that I circled in that Liver Down Under, he published this editorial. He suggested liver stem cells, where art thou? He actually challenged the concept of a liver stem cell. And this was a problem for me because, one, a lot of my collaborators were in Melbourne. My cells are tumorigenic, given cancer, and liver stem cells, existence of which is now questioned. And we know in the grant system, they need a reason not to fund you. And these are 2 reasons not to fund me. So I had a difficulty in getting funding from henceforth. All right, so current projects. I'll tell you about the Bustleton Jetty and the biota pipeline. What I did was I changed my emphasis. Instead of now using these cells for therapy, I've got cancer liver stem cells, and I've got non-cancerous liver stem cells. One smiles and gives good hepatocytes, and one is frowning and gives hepatoma, liver cancer. What we're doing now is actually using these 2 types of cells to test in the biota pipeline. This farmer, Chris Rickstein, has provided funding to the Harry Perkins to screen our natural products for anti-cancer agents. What I can bring to the party is to screen for agents that not only kill the cancer cell, but spares the normal cell. So it's selective. So it's a tall order we are asking, but it's a very, very important one. And the Bustleton Jetty marine sponge, if you go on their website, you'll see this undertaking, and it's initiated and supported by Lisa Shreve, who's the CEO there, and Danielle Dowding. So we're undertaking these studies now, so I've come to what I'm doing today. So here's the rotten apple, which are my cancerous liver stem cells, and here's the good apple, good guys. And what we're doing is we're screening for agents that kill this rotten apple and spare the good one. That's what we're heading towards, all right? What have we done? Bustleton Jetty has already collected 30 sponges. That's complete. I told them to put my name on that little bag, but they didn't. Never mind. But that's important, that they are now harvesting these. They've gone diving, and we grind them up, we extract them, and Felicia, who is in my lab, is using a robot and microscopes that are inside these incubators, and these microscopes are taking pictures of these cells every 6 hours, and they can go on for weeks. And from that, we can plot a growth curve or a death curve for the cells. So we're monitoring these cells. And these pictures can come up on your iPhone. So if you don't come, you can have a look at your results even when you're not at work. So a great facility made available by the resources of this wonderful organization that I'm proud to be part of. So we're doing this right now, and we're screening. So work is proceeding in earnest. The future projects, the biota pipeline will expand. There are 226 other sponges from the WA Museum collection, 5 sea cucumbers, native plants, over 500. And I think some are from Kingsley Dixon, who recently had some award. He was the person working at the Kings Park laboratories. And we also will have native plants from indigenous owners, the Bardi territories. Then if we find something useful for our liver cancer screen, we can test this extract on other types of cancers. And this work is going with Gavin Flamati, who is a chemist on campus, Brodie Bulcock, who we've hired, and Felicia is working on the screening that we're doing here, and the WA Museum is also a partner. So I'm going to close by telling you about a current project, which is cystic fibrosis. I know very little about cystic fibrosis. A colleague of mine told me, did you realize, George, that cystic fibrotic kids can have a liver problem as well, a comorbidity? 1 in 2,500 newborns in Australia have cystic fibrosis. It's very common. 10% have liver-associated liver disease, and that's LD. So we need to diagnose cystic fibrotic liver disease children. Why? The liver disease may complicate the medication that's given to the cystic fibrotic kids. Second, we need to detect it early because the liver condition can be mitigated by intervention, giving them better diets that don't damage their liver, etc., etc. Currently, they are diagnosed by a liver transaminase, which is not reliable. Very briefly, the transaminases are there in the blood when the liver cell breaks up and spills contents into the blood, then you can pick it up. But if the liver cell is sick and not breaking up, it doesn't necessarily shed the liver enzymes into your blood. They're not reliable, and they cannot detect early changes in the liver status. The gold standard is to put a needle into the cystic fibrotic kids, pull out a bit of liver, and have a look at it. Now, that's not trivial. And the biopsy is only a tiny bit of the liver. You may miss areas of importance. It does not represent the whole liver. So what we're looking for is what I call the canary in the coal mine. So we hypothesize in the liver of children with cystic fibrosis, like the alcoholics, like the genetic hemochromatosis, like the hepatitis, they have liver stem cells because their liver is not well. That's the first thing. And we're also hypothesizing that the numbers of these liver stem cells reflect the severity of the liver disease. And here, the collaborators in this project, Ingrid Lang, who's in the audience, and Marcello Leal, who is the gastroenterologist at the Children's Hospital. They alerted me to how my expertise in the liver might be able to help these children. We also hypothesized that in the blood, that the children with cystic fibrosis will have bits of the liver stem cells. The analogy with the canary is maybe bits of the feathers of the canary is in the blood of these kids. And if we can detect and to quantify this biomarker, we can then diagnose and reveal the severity of the liver disease, and we may pick it up early. A lot of work to do still. But we are very fortunate. We receive funding to do this work. And the people who are carrying on with this they are in the audience. Augustine Seguro and Adam Passman are both working on this. So our first step is to look for the canary. And indeed, we do find the canary is there. Remember I told you about the blue cells and the brown cells? In mild liver disease, mostly blue cells. But in moderate liver disease, we still start to see some brown cells. And in severe, lots and lots of brown cells. These are the liver stem cells. So what we're saying is, in the cystic fibrotic kids with liver disease, they have liver stem cells in their liver, okay? And we get some sort of a correlation between the severity of disease and the numbers of these stem cells. But as I said, it's not trivial to take a liver biopsy. So we are looking for the feathers. So our second step is that we are looking for the canary's feathers. But at the moment, the results that we have are bewildering. They're very puzzling. We've developed an assay using that monoclonal antibody that we got from Martin Pera and from the Australian Stem Cell Centre. We can find the canary, but we can't find the feathers in the blood. We have used a commercial antibody, and there's a trace of something in the blood. So that's where we're at. So we have a puzzle to solve. And our proposal about how to solve this puzzle is that we are now thinking along the lines that only part of the feather is in the blood, and we need to discover which bit is there. So here's the canary, here's the feather. We can see both of these, but when in the blood, we don't know which part of the feather is in the blood. So we need to find the right antibody to detect both the canary and a bit of the feather in the blood. So I've come around a full circle. I came to this country, I spoke broken English, I had no family, no house, no car. I've got all of that. And it's through the generosity of all you people here. And I am deeply, deeply indebted. Very, very, very grateful for what I have, right? And look at this. You know, I've got a family here. You know. So, I mean, I know that try as I might, I'm not ever going to be able to balance this ledger, but I will keep trying. Thank you for your attention.